Have you ever wondered why some animals can regrow their limbs while humans can't? This question has intrigued scientists for years, leading them down the fascinating path of limb regeneration research. Certain creatures on our planet, like starfish and newts, possess an incredible ability. They can regrow lost limbs almost as if by magic, but it's not magic, it's biology, at its most mind-boggling. These animals, when injured, trigger a process called blastema formation, setting in motion a series of biological events that result in a fully formed, functional limb. Imagine a starfish losing one of its arms. Instead of being permanently impaired, it merely grows another. Or consider a newt whose tail is snatched by a predator. This newt doesn't fret. Instead, it regenerates a new tail, as good as the original. This process isn't just about growing new body parts. It's about restoring full functionality, complete with nerves, muscles, and blood vessels. But why should we care about starfish and newts? Well, understanding this process has significant implications for medical science, especially for humans who lose limbs due to accidents, disease, or birth defects. Right now, the best we can offer these individuals are prosthetics, artificial limbs that can help restore some functionality, but never quite replicate the real thing. But imagine if we could harness the power of regeneration, if we could trigger blastema formation in humans. This isn't just the stuff of science fiction. Researchers are already making strides in this direction. They're studying these amazing animals, working to unlock the secrets of their regeneration abilities. They're exploring the genetics, the cellular mechanisms, the signals that trigger and guide this process. These discoveries could pave the way for revolutionary treatments. We could potentially regrow lost limbs, repair damaged organs, and restore functionality in a way that prosthetics and surgeries can only dream of. This is the promise of regenerative medicine, a field that's still in its infancy but holds immense potential. If we can unlock the secrets behind this process, we could potentially revolutionize medicine. And that, dear listeners, is the wonder of limb regeneration. To understand limb regeneration, we need to delve into a process called denervation. This term refers to the loss of nerve supply to a muscle, and it plays a significant role in the functionality of that muscle. Now, the relationship between denervation and muscle function is a time-sensitive one. The longer a muscle is denervated, the more it affects both the muscle and the motor nerves that were once connected to it. Picture this: when a muscle is denervated, it doesn't just sit idle. It undergoes significant transformations. These changes include ionic imbalances, decreased resting membrane potential, accelerated protein breakdown, and even the activation of the intracellular inflammasome, a complex of intracellular proteins that trigger inflammation. As the denervation period extends, the muscle's contractile apparatus, the components that enable muscle contraction, progressively disintegrates. The number of mitochondria within the muscle decreases, leading to a substantial decline in contractility, the muscle's ability to contract. Simultaneously, the muscle's capillary bed, which supplies it with oxygen and nutrients, degenerates. This creates a hypoxic or low oxygen environment that promotes the growth of fibrous tissue, further inhibiting muscle function. The result? Long-term denervated muscles exhibit a reduced number of muscle fibers, profound atrophy, and a surrounding dense meshwork of collagenous fibers and fat cells. Historically, this state has been considered irreversible and permanent. However, recent evidence challenges this notion, suggesting that some of these degenerative changes may be partially reversible with long-term external electrical muscle stimulation programs. But here's the catch: it's not that the chronically denervated muscle fibers can't accept reinnervation or the restoration of nerve supply. Rather, the remaining intramuscular neural pathways, which persist even after the degeneration of native nerve fibers, become less conducive for regenerating nerve fibers. The effects of denervation are profound and significantly impact the potential for successful muscular function restoration. But understanding these effects is crucial for advancing our capabilities in limb regeneration. One of the major outcomes of denervation is what we call Denov atrophy. This is a process characterized by the progressive remodeling of the denervated musculature and is often considered the main factor in poor functional recovery after peripheral nerve injuries. Now, let's delve into what happens at a cellular level. After denervation, muscle fibers undergo profound changes. An ionic imbalance occurs, leading to a decreased resting membrane potential. This means the cell's normal idle state or resting state is disrupted, Additionally, protein catabolism, or the breakdown of proteins into simpler compounds, accelerates. This rapid protein breakdown can lead to muscle weakness and wasting, but that's not all. The sarcolemma, or muscle cell membrane, becomes more permeable, making the cell more vulnerable to substances that could potentially harm it. And within the cell, the intracellular inflammasome, a complex set of proteins that regulate inflammation, is activated. As time goes on, the contractile apparatus, the machinery of the muscle cell that allows it to contract, progressively disintegrates. The number of intramuscular mitochondria, the powerhouses of the cell, decreases. The result of these changes is a substantial decline in contractility, the ability of the muscle to contract and generate force. Concurrently, the intramuscular capillary bed degenerates, leading to a hypoxic or oxygen-starved intramuscular environment. This lack of oxygen triggers fibrotic remodeling, where healthy muscle tissue is replaced with fibrous connective tissue. This can further exacerbate muscle weakness and loss of function. Long-term denervated muscles typically exhibit a reduced number of muscle fibers with profound atrophy. They possess a disorganized contractile apparatus and are surrounded by a dense meshwork of collagenous fibers and adipocytes, or fat cells. Historically, this state of affairs has been seen as irreversible, with a permanent loss of function. However, recent evidence suggests that aspects of these degenerative changes may be partly reversible under certain conditions. For example, long-term external electrical muscle stimulation programs have shown promise in reversing some of this damage. Denov atrophy, while often seen as irreversible, may be partly reversible under certain conditions. So while the journey might be tough, there is a glimmer of hope for our denervated muscles. Reinnovating long-term denervated muscles is a challenge in itself. The journey is fraught with difficulties, largely due to the profound cellular changes that denervated muscle fibers undergo. These changes include ionic imbalance, decreased resting membrane potential, accelerated protein catabolism, permeabilization of the sarcolemma, and activation of the intracellular inflammasome. As the denervation time increases, the muscle's contractile apparatus progressively disintegrates and the number of intramuscular mitochondria decreases. This results in a substantial decline in contractility. Simultaneously, the intramuscular capillary bed degenerates, leading to a hypoxic intramuscular environment and fibrotic remodeling. Over time, denervated muscles typically exhibit a reduced number of muscle fibers with profound atrophy, a disorganized contractile apparatus, and a surrounding dense meshwork of collagenous fibers and adipocytes. Historically, this state was considered irreversible, with a permanent loss of function. However, recent evidence challenges this notion. Some aspects of these degenerative changes seem to be partly reversible by long-term external electrical muscle stimulation programs. Occasionally, functional recovery has even been observed after long periods of denervation, but attempts to re-innovate long-term denervated muscles are generally less successful. It's not that the chronically denervated muscle fibers cannot accept re-innovation. The key factor limiting success is that the remaining intramuscular neural pathways, which persist after the degeneration of native nerve fibers, become less conducive for regenerating nerve fibers. Now let's talk about prolonged distal pathway denervation. After nerve injury, endoneurial tubes within denervated nerve segments serve as guiding structures for regenerating axons and provide a supportive environment through proliferating Schwann cells. However, with prolonged axonal deprivation, these Schwann cells atrophy and the endoneurial tubes become progressively occluded by collagen filaments. Collectively, these changes can reduce the number of injured motor neurons that successfully regenerate their axons to less than 10%. The process of denervation and the subsequent challenges of re-innovation are complex, but understanding them is key to unlocking the potential of limb regeneration. Today, we've taken a deep dive into the world of limb regeneration and denervation. We've explored the fascinating yet complex processes of denervation atrophy and the challenges of re-innovation. We've seen how denervation can impact muscle function and how this understanding is crucial in the realm of limb regeneration. The journey towards achieving full limb regeneration is fraught with complexities, but every new piece of knowledge brings us closer. While we may not be able to regrow limbs just yet, understanding these processes brings us one step closer to making that a reality.